Blue-white selection is basically a screening method for screening the recombinants. Now, bacteria generally take up plasmid in the wild. Not only plasmid, it can take up any naked DNA in the natural environment, and that is called as a natural transformation. But the same method can also be employed in a laboratory to transfer the vector DNA into these bacterial cells. Here on the screen you can see there is a plasmid DNA, and it has been transformed inside a bacterial DNA. And here there are two DNAs now. One is the bacterial chromosome, and the other smaller red circular DNA that you can see, it is the plasmid DNA. So these cells are said to be transformed with a particular plasmid. Now, the plasmids that we are using, they must have certain characteristics. Also, the genome or the bigger DNA that you can see over here, that is the bacterial genome, they should also be modified with specific changes so that the plasmid is able to survive inside the host, plasmid is able to express, and the expression of the plasmid is not masked by the expression of the genomic DNA of the host. So I will discuss that in detail. First of all, which kinds of plasmid vectors are suitable? So there are a few properties which are essential. First one is replication. The plasmid that is entering inside the host should be able to multiply, should be able to replicate. And for replication, origin of replication, the sequence of DNA which is the initiating sequence where all the proteins for replication, they identify that particular sequence and begin the replication of DNA. So that sequence must be there on the plasmid. Hence, if you can see over here, this is a PUC19 vector. You can see there is PMB1 ori over here. Next is the selectable marker. Now, selectable marker is any DNA sequence which codes for a particular protein, and because of that protein, you are able to confer a selection property. Now here you can see this green sequence which is written AMP. Now this is basically a region which produces a protein that can break the antibiotic ampicillin. So if this plasmid is present inside the host, then this protein which is resistant to ampicillin, which can break ampicillin, will be produced, and because of the production of this protein, the bacteria will be able to will be able to destroy the antibiotic, and hence it will survive. Again, this is very important property. Then is the restriction sites. Now restriction sites are essential because we use these plasmid vectors for inserting a particular gene of interest and then storing them, maybe for the purpose of libraries, or we might want to express a particular gene. So in either of the cases, we have to first insert it in a site which can be restricted using these restriction enzymes. So here you can see MCS. This is a multiple cloning site, and a multiple cloning site is where there are recognition sequences for different restriction enzymes. You can see there's a whole list of restriction enzymes which can be used. Now, this also helps the researcher or the person who's working in the lab, because maybe it is not, maybe all the enzymes may not be available. Or if imagine if you have only one particular restriction site and you do not have that particular enzyme, then you are in a dilemma of whether to buy a new enzyme, and then enzymes might be expensive and so on and so forth. So if you have options of using multiple enzymes, then you can choose out of this entire list, you can choose just one enzyme which is already available in your lab, and you can use that. So the restriction sites are basically used for breaking open this circular DNA And when it is broken, then the gene of interest can be incorporated within this particular region. Then a suitable host. Now, as I told you before, a suitable host is necessary. A host which can permit the replication of this particular plasmid vector, a host which does not mask the expression. From masking the expression, what I meant is, suppose your plasmid is producing an ampicillin resistant protein, and your genome, the host genome, is also producing the ampicillin resistant protein. Then, if you try to select it on an ampicillin antibiotic plate, you will not be able to differentiate between which one has the plasmid, which particular cell has the plasmid, which particular cell doesn't have the plasmid. So, if your plasmid has ampicillin resistance marker, then your host should be ampicillin amp minus. That means it does not carry this particular, it should not carry this particular gene. I hope I am very clear in this, that if there is a particular gene which is producing a particular protein for the purpose of selection is produced by a plasmid, then it should not be produced by the host. Next, the lacz gene. If you can see over here in blue, there's a lacz marker. Now this lacz marker is in two parts. It is broken into two parts. So a part of this lacz, so lacz basically encodes for a beta-galactosidase protein, and it is in two parts. One part which is produced by the vector, the other part which is produced from the genome of the host. So how do you make it a suitable host? That the suitable host will produce one part, and the other part is produced by the lacz marker. So when they come together, they form a complex, and that complex then becomes an active beta galactosidase protein. Next is high copy number. That means the plasmids which are entering inside the host, they should be able to multiply and should reach a particular number which is fairly high, so that when you want to harvest the plasmid DNA, whenever you want to work with the DNA, you get sufficient amount of yield in order to perform your DNA manipulations. Now, this lacz, if we insert a particular gene of interest at this MCS, then this lacz gene gets disrupted. And because of the disruption of this lacz, a functional polypeptide will not be produced. If there is no insertion in this MCS, then a functional polypeptide of lacz, that is a part of beta galactosidase that I told you, that will be produced. So it totally depends upon the insertion of the gene of interest, whether your lacz will be producing a functional polypeptide or not. Now, when this plasmid, imagine that you have inserted a particular gene of interest inside this MCS, multiple cloning site, and now you have transformed it into the bacteria. Now you are performing all of this in a tube, so you do not know which bacteria has taken up. First of all, you do not know which plasmid has the gene of interest and which doesn't. You are expecting that most of them have. Secondly, when you are transforming them inside the cell, you do not know which one has taken up the the plasmid with the gene of interest, and which ones have taken up the plasmid without the gene of interest. Or there can be a wrong insertion also. So let's go ahead and check. So this is the plasmid vector and this is the foreign DNA. Now you try to ligate them together. When you ligate them together, there can be three possibilities. One, that it gets ligated at the right site within the MCS and it disrupts the lacz. The other could be an insertion where it is not within the lacz but outside the lacz. And the third, that there is no insert, that the lacz, the foreign DNA, it did not insert itself into the vector. Now, all these three cases will have three different outcomes. In the first case, where lacz was disrupted, you will see that there will be white colonies which will be formed when you perform the XGAL confirmation, the screening using IPTG and XGAL. Why white colonies? Because here the laxi were disrupted. Laxi will not be producing a functional polypeptide. If there is no functional polypeptide, then it will not make the complex which will ultimately hydrolyze this X-gal, which is a chromogenic substrate, and give blue color. So you do not get blue color, you get white colonies. In the next case, there is an insertion which is not inside the laxi, and hence laxi is normal and it will be able to form the complex, and you will see that it will hydrolyze the X-gal also and produce a blue color. So you expect that they will get blue colonies in this case. When there is no insert, so no insert, no harm to the laxi, it is not disrupted. So again, laxi will be produced and it will be able to form the complex with its counterpart on the genome, and again it will hydrolyze the X-gal and it will produce blue colonies. So this is how a plate on which you have used for the screening will look like. You can see that there are white colonies and blue colonies. The white colonies are essentially the colonies which have the gene of interest inserted within the multiple cloning site of the vector, and the blue colonies are the ones which either have inserted the gene of interest at a wrong place, or they do not have the gene of interest inserted in the vector at all. So in either of the cases, you do not need these particular cells. You need these white cells. These white colonies are the ones which are carrying the gene of interest. So you can take a loop full from this plate from these white colonies, culture them in the presence of antibiotics so that sufficient pressure is exerted. Especially, I think this experiment was done with the help of PUC-19, so you can use ampicillin in this case on the plate as well as while culturing. So when you provide a sufficient pressure of the antibiotic, a sufficient concentration of 1 microgram per microliter is the concentration that we generally use. So then you will see that the plasmid is maintained inside the cells, and this because cells cannot afford to lose that particular plasmid. If they lose the plasmid, they will die in presence of the ampicillin. So I hope you understood what is blue white selection, how are these cells selected, and how can we screen our cells with the gene of interest. Thank you very much.